It's the first time I've been to UAB. It's clearly a really vibrant, lively place, and I'm happy to see that you folks are interested in systems biology. So, being the first speaker, I assumed that you were relying on me to actually define what systems biology is. If I try to do that with words, I'll be doing that until the end of the hour. So instead, I thought it would be better to do it with pictures, pictures being worth 1,000 words. So this was biology before systems biology. This was the epidermal growth factor receptor pathway about 25 years ago. This is the way we all learned it, and in fact, this is the way we tend to teach our graduate students to this day. But of course, you know, this is the epidermal growth factor pathway about 5 years ago, and in the last 5 years, it's gotten even more complicated than that. This is what genetic interactions look like nowadays with connections all over the place. This is what protein-protein interactions look like. And so basically, systems biology is about the fact that this is what biology looks like, right? Everything is one big hairball. And the goal of systems biology is to make sense of the hairball. And that's certainly not the way that most of us were trained. You know, certainly when I was a graduate student, it was more of a one-at-a-time thing. And now we're all being forced to look at it from a much more of a 30,000-foot view and say, you know, what really is going on in this sort of thing. Okay, so that's really the question of what do you do about the complexity of biology? Once you've realized you can't ignore it anymore, right, how do you deal with it? And there's really two ways to deal with it, right? One is figuring out how do you get from hairballs to predictions, right? How do you take that stuff that just looks like a mess of interacting behaviors and draw useful inferences, right, directly from the data. And thank you. The middle one, yeah. Okay. And that's something that I think the other speakers in the symposium today are going to tell you a lot about this side of systems biology. But there's the other side of systems biology, which is the side that I tend to work mostly on, which is how do you understand why those hairballs are there in the first place? Right? What's the reasons behind these hairballs? Do they actually have structure? Is there some reason why biology is organized this way, and can we actually understand it? So that's the problem of getting to why, right? Why are things the way they are? And you know, there's a long tradition of people arguing that there's no point in trying to get to why because maybe biology is just complicated by chance. Right? Maybe it's just kludgy, ill-designed, right? And so, you know, rather famous people like Stephen Jay Gould have been known to write things like, you know, if some fish hadn't by accident evolved fins that could bear weight, we never would have had terrestrial vertebrates. So we're glorious accidents of an unpredictable process with no drive to complexity, not the expected results of principles that yearn to produce a creature capable of understanding the mode of its own necessary construction. This has been a very popular view in biology, that really we can avoid paying attention to the complexity of it all because it's just there by chance. But of course there is another view that biology is not complicated by chance, that it's complicated by necessity. So here's something else that's a hairball. Basically, if you looked at the wiring diagram of this cockpit of the 787 Dreamliner, it would look just as much like a hairball as any biological system. But no one would argue that it's this way by chance, right? It's this way by necessity, right? So the plane can actually do its job. So what we learned from engineered systems is these systems have to perform robustly. So what that means is that the world is an uncertain place, right? We don't know if there's going to be turbulence right now or rain or birds flying into the engines or any number of things. And so you have to be able to control how you behave, right? So that you get the performance you want despite the unpredictability of the world. And control is a big driver of complexity. And the reason why that's the case is because control always comes at a price, right? You never get control for free, and that can be illustrated by what engineers like to call the no free lunch principle, right? Achieving good performance on anything always comes at the expense of good performance in something else. That's the notion of trade-offs. So a good example in engineering is, you know, this is a really fuel-efficient car, and it's the worst one to be in if you're going to have a crash. This is probably the best one to be in if you're going to have a crash, and of course it's the least fuel efficient, right? It's no accident that there are these trade-offs. And of course, you see exactly the same trade-offs in biological systems, right? These have the best ability to control body heat, but they don't fly very well, right? And these things fly beautifully, but they lose heat like crazy, right? So there's these relationships all over. And so that enables us to pose the question, to what extent do the constraints imposed by the need for control, and particularly to control for many uncertainties at once, which biological systems face just as much as engineering systems do, to what extent does this give you the structures, the complex hairballs that we see in biology? To what extent can we really make sense of them in terms of the constraints that control objectives place on them? Okay, so much for sort of the 150,000 foot view. Now we're going to descend a little bit lower, and we're going to talk about some specific systems and specific problems. And first I'm going to talk about tissue growth and homeostasis, and that's going to be related directly to issues of development, regeneration, and cancer. Right? And in that arena, I'm going to talk about issues of control and how we can use our knowledge about what needs to be controlled to try to make sense of what looks like a very small hairball, not even a really big one, but just a few interactions that otherwise might look rather random and arbitrary. And then in the last sort of third of my talk, I'm going to move on to the larger, more general topic of complex genetics and ask whether there are issues of control within the world of genetics, right? Controlling the effects of mutations in our genes that have an impact on the way genetic interactions become structured. Okay? So that's the basic outline of what I'm going to talk about. And the first part is about tissue growth and homeostasis. All right. So we'll begin, rather than by talking about examples or molecules, talking about just patterns of organization. What do we see when we look at the systems that biological organisms use to generate tissues and maintain them and so on? We see the use of stem cells, cell lineages. We see that cell types are segregated in particular spatial ways. We see a common use of morphology called epithelial. We see feedback, which we'll talk about in a little bit. We see that mechanics affects growth. We see that there's finite numbers of divisions that cells tend to go through, that there are lifespans. And we see that in just about every species, cancers occur. And we see that cancers maintain lineage structure. So this is a somewhat more precise way of articulating what people refer to as the cancer stem cell hypothesis. The hypothesis that a cancer is not one cell gone awry that just does what it wants, but rather the lineage progression that normally goes on in a tissue from stem to differentiated continues to go on in an aberrant way in cancers. So these are all things we see. And then we can say, what are some of the control objectives? What are the things that tissues need to control? And so, for example, tissues need to be the right size. Right? Our tissues have an amazing ability to be the right size. Right? Look at your two arms. They come out the same size even though these cells stopped talking to those cells back when you were at gastrulation. Right? How is that possible, right? That you get such precision of size? And other tissues like your skin, it knows how to be thick enough, right? Not too thick, not too thin, and not making you look like a Shar-Pei dog, right? So there are all these controls that are clearly going on the level of size. And they relate to size fluctuation as well in tissues that turn over, right? They maintain a constant size. They're not doing this all the time. And of course, you have to produce different cell types and at the right time and in the right places, in the right proportions. And oftentimes, the size of a tissue relates to the size of another tissue, right? The liver may be related to the size of the spleen and so on, and some of these change size together. You need to regenerate rapidly after injury, right? So you have to come back faster than you might be turning over a tissue normally. And you have to deal with the effects, with the fact that you pick up mutations. There's a real mutation rate every time cells divide. And we know what happens when you get somatic mutations, right? You get a risk of cancer and a risk of passing mutations on to the next generation. And finally, tissues need to carry out morphogenesis. They need to create shape and form. So that's a lot of control objectives, right? And all of it has to work. So the question is, how is it done, and how does doing any one thing interfere with something else so that doing all of it requires a lot of complexity. So we'll begin with the issue of just controlling growth. What's so hard about controlling growth? This is something so simple we often don't think about it, right? Is that the growth of cells is like a nuclear reactor, right? It's an autocatalytic process. Cells make cells, which then make cells, right? Mathematically, that's an exponential explosion process. The fact that it's controlled at all is remarkable, right? Think of how hard it is to control a nuclear reactor, right? And get an even output of energy from it, right? It's not trivial. You can't just change a little rate constant here and have the whole thing come into control. It wants to explode. So for example, in a tissue like your brain that doesn't renew, but you just have to make a certain size and stop, right? How can you do that with an exponential process, an explosion at the foundation of that? Well, the simplest thing you can do is you just explode and then you shut it off at a certain time point, right? You just count cell cycles. Okay. So is that really a viable strategy? Well, we know from genetics of mice that the brains of genetically identical organisms vary in size within 5% of each other. Right? So in order to do that, given the expansion allotted, you'd have to have cell cycles accurate within 1 part in 1,000. And we know cells don't come close to that in terms of the variation in their cell cycle length. Right? So how do you achieve that kind of precision? And likewise, in a tissue that's continually turning over, right, the only way to achieve stability is if the cells die at exactly the same rate that they divide. Right? They divide a certain period of time to divide and a certain period of time to die, and they must be exactly balanced, or else the tissue will explode or shrink down to nothing and vanish. Right? So how do you achieve that kind of exact precision in biology? So the notion of stem cells or stem lineages, the idea that you have a cell at the base of a lineage that does the dividing and then produces a terminal cell, seems like one way to solve the problem. Because, of course, when you have that, stem cells divide, they make one of themselves and one of those, and so these are now produced at a constant rate, not an exponential rate. Right? These terminal cells are being produced linearly. And so any death rate will cause this system to come to an equilibrium, right, to a steady state where you have some constant number of cells. But of course, the problem is that introduces a constraint, which is somehow the stem cells have to know when they divide to make one differentiated cell and one of themselves. Right? They got to know to do that, because if they make two differentiated cells some of the time, or two of the terminal cells some of the time, then you wouldn't have constancy. Okay? So if you look in most textbooks and then you look at what we teach graduate students, we say, well, that problem is solved because stem cells are special cells. They know how to do what's called asymmetric division, right? They divide and one cell naturally becomes something else and one cell remains a stem cell, right? That's considered the kind of hallmark of stem cells. But the problem is that, which is a control strategy in its own right, comes at a price. And the price has to do with the accumulation of mutations. Right? So if you have a tissue that's turning over hundreds, thousands of cell cycles in your lifetime, like say your intestinal crypt, you're constantly picking up mutations. We know this is a fact, that you're picking up mutations. And if you have this type of a lineage structure, that every time you pick up a mutation, you keep it. And then when you pick up a new mutation, it happens on the background of an old mutation, and you eventually accumulate a lot of mutations. And that, of course, is going to increase your risk of bad things happening like cancer. Now, you might think that having a different pattern of division actually could be helpful. So, for example, if instead of every stem cell making one of these and one of those, sometimes it makes two stem cells and sometimes it makes no stem cells, right? And as long as those two things are balanced, you can still have the same final output. But now some of these mutations get swept away, right? So this one appears, but then it's gone. This one appears and then it's gone. So you think, well, division pattern actually could be a way to get around this. But then if you think a little more, you go, well, wait a minute. For every time this happens, which is good, this will happen, which is extra bad, right? Which is the cell that has a mutation will make two stem cells. And now you have twice the probability that this new mutation will land in something that had a mutation. So your kind of initial thought would be, well, these two things will just balance each other out. There's really no benefit in dividing symmetrically rather than asymmetrically, but that turns out actually not to be the case because of the mathematics of stochastic processes, basically where individual things are happening noisily. What you find if you actually calculate by doing many, many simulations, that if you have a high degree of symmetry in your stem cell divisions, right, then the number of cells that have achieved 2 mutations at a certain period of time goes down the more symmetric your divisions are, and it can be up to 100-fold. And the reason for that is because in this little game of mutation accumulation, what happens if stem cells divide symmetrically is clones of stem cells randomly fluctuate up and fluctuate down and fluctuate up and then extinguish every now and then. And most of the time they extinguish after a certain period of time. And so the only way to get 2 mutations is if you happen to hit a cell before hit a clone that has a mutation before that clone goes extinct. So you have kind of a timing game. And if you can space those timings out, then it's very likely that 2 bad things will happen in the same time interval. And you can get a tremendous amount of protection. In fact, this works most strongly when the mutation rate is accelerating, as it is, for example, when the first mutation creates a genetic instability, as is often the case in cancer. Okay. So I brought all this up from a purely theoretical point of view, but obviously I wouldn't be telling you this if it weren't the case that stem cells do divide symmetrically a lot, despite what textbooks say or used to say. In your skin, the stem cells are dividing symmetrically 16% of the time, and in your intestinal crypts, which are the cells faced with the highest mutation load that we know so far from studies that have been done in animals, 80% of the divisions are symmetric divisions, right? So that classic asymmetric behavior of stem cells kind of out the window for those sorts of tissues. Okay, so symmetric division helps you deal with the problem of mutation accumulation, but now it comes at a price. Because now to achieve any sort of steady state, you have to exactly balance how many times stem cells make 2 stem cells with how many times stem cells make no stem cells, right? You have to bring this into balance, otherwise populations will always explode or extinguish. And that suggests there must be some sort of feedback in the system that's used to bring it into balance. And in fact, there has been evidence for feedback in tissue growth control for ages. Back even in the 1960s, when you can get away with publishing a review in Cancer Research with the word speculative in the title, which I don't think you could do anymore. But the term kalone was coined as a feedback factor, a secreted feedback factor that tissues would produce that would slow their growth, right, and bring them into control. It wasn't until the 90s that the first kalone was actually identified. It's called GDF8 or myostatin. It's produced by muscle, and it feeds back and inhibits the production of muscle from muscle stem cells. Okay, and so GDF8 or myostatin, this is a mouse that has a myostatin mutation, and it has twice as much muscle. And it's actually quite strong. This is the Belgian blue cattle. It's supposed to make really good eating. I don't think I've ever had one of these steaks, but they're popular in Europe, and that's a GDF8 mutation as well. There are people with GDF8 mutations, and I don't mean Arnold Schwarzenegger. That's pharmacology, that's different, okay? So how might this kind of feedback work? So the traditional view in the kind of early days of kalone biology was you feed back on the speed at which stem cells divide, right? If you have too much tissue, you divide slower. Too little tissue, you divide faster. The problem is you can actually do the math on that. It's very simple, and it doesn't really work very well. First of all, it doesn't solve the problem that the stem cells still need to know to apportion their divisions exactly equally, so that for every stem cell division, on average, you make just 1 stem cell, even though the individual stem cells are quite stochastic. It doesn't provide robustness, the idea that you want to keep something at a constant, even level, despite the fact that the environment is varying. And if you have this little feedback loop in a system like this, and say you change the rate at which cells die, this feedback loop helps a little bit to keep this constant, but not a whole lot. And it doesn't give you another objective that we talked about, which is regenerating fast. If you have a system like this and you say take away the terminal cells and you let it come back, it does come back a bit faster, maybe even 2-fold faster if you're lucky, but that's about it. Whereas our real tissues, you can have tissues where the cells aren't turning over very fast at all, and you injure them, and within a day they're now turning over 5 or 6 times the rate, right? That's very typical of our tissues. So this kind of feedback structure doesn't work that well. It turns out a different feedback structure works better. And so I have this V and D, which previously represented the rate at which cells divide and the rate at which they die. And now I'm going to introduce this thing called P, which is the probability that the progeny of this cell go back to becoming that cell. So that's the thing that has to be 1/2, right, if stem cells are going to maintain constancy, right? And so if you actually feed back on that probability, so that probability is adjustable depending on how much tissue you make, you can write that down in some very simple equations. But the interesting thing is now you get automatic homeostasis. The stem cell populations always achieve a 50-50 split, independent of what each cell does and independent of the details of how the feedback is implemented. In fact, you get what engineers call perfect robustness. Here's just a little toy model of that basic system in which you just take away all the terminal cells and you allow it to regenerate back. And it doesn't matter how many cells you start with, how fast they turn over, how fast they divide, they regenerate right back to the same number. Okay? And the reason they can do that, they have this perfect robustness, is because they're implementing something engineers call integral feedback control. And if, just to be very quick about what that means, integral feedback control is when you have a feedback system where the error, the difference between where it is and where you want it to be, right, accumulates over time. It gets worse and worse and worse and worse until you get to where you're supposed to be. Those kinds of integral control systems drive things right to the set point. And that's exactly what this accomplishes. Unless the terminal cells are exactly the right number, stem cells are out of control and go in the other direction until they get it back to that number. And so you always end up with that half. So you don't need asymmetric divisions at all. In fact, you can have really messy, ugly systems with lots of math flying around, and because of this integral feedback, the number of cells you get at the end are all just determined by that one little feedback loop, which is a nice thing. That's what you want in a system, to make it completely robust to all kinds of uncertainty in the world and just have one thing that you have to control, which is the strength of the feedback gain. And in fact, that simple strategy explains things like how you can make brains with such small coefficients of variation. Because if you try to build it without feedback and you start with half as many brain cells to begin with, you get half as big a brain. Not very helpful. But if you do the same system but you have this feedback loop, then it doesn't matter how many cells you start with to found your brain, you still end up with a brain of the same size. Okay, so a lot of power in that particular feedback loop. And again, I wouldn't have been introducing it if I didn't know that that was what actually happens in vivo. So we in fact know in the case of GDF8 that that's how it works. It doesn't feed back particularly on the speed at which muscle progenitors divide. It feeds back on the probability that their offspring will become myocytes. In the olfactory epithelium, which is a system we've worked on, GDF11, which is a homologue of GDF8, and activin, which is also in the same family, both feed back onto p-values, these renewal probabilities in this system. And in the hemopoietic system, where we don't quite know the identities of all the factors yet, but we know there are negative feedback factors that in fact do exactly the same thing. They target these renewal probabilities so that you can keep these systems under control. Now, one of those systems I mentioned, the olfactory epithelium, is a bit more complicated because you have two stages. You're feeding back on a middle stage and feeding back on the stem cell stage, right? And this is a system that makes neurons in the back of your nose so you can smell your good food. And so one of the questions is, well, what is this doing for you? And there's some interesting behaviors that are very useful that come out of feeding back on an intermediate stage. And those are things like when you ask these systems to regenerate by taking the final stage away, they come roaring back, right? And they come roaring back in a particular way where the precursors come up and then go down transiently, right? And that's actually what you see in most regenerating systems. The stem and progenitor cells expand and then contract, and then you go back to normal as they pump out a lot of differentiated cells at a very fast rate. And also, this feedback loop can give you a lot of robustness, right? You can get the right size tissue no matter how you vary what you put in here. But there's another trade-off issue, which is you can't do both things at the same time. That is, you can configure the system to be robust but not fast, or you can configure the system to be fast but not robust, but there's no way to configure that system to do both things simultaneously. That's that notion of control trade-offs. This is just a figure showing that by exploration of parameters that you can either be up here or over here, but you can't be over there, where you're both fast and robust. Now, also there's some other problems that the speed of regeneration depends exactly on the initial conditions in a way you don't really want it to. Now, one way you can get around that is by merging two negative feedback loops together and relying on this integral feedback loop and this feedback loop. But then that still begs another question, which is that the integral feedback itself comes at a price. Okay, remember, everything has a price. And in this case, the price is that even though this system has perfect robustness with respect to this or how many stem cells you make or any number of other things, it's completely fragile, that's the opposite of robust, fragile, to perturbations that happen in the feedback itself. In other words, if you were to knock out the feedback factor that goes back to the stem cell, you would predict the system would explode, right? So fortunately in biology we can do experiments and the system doesn't explode. We actually know that the mouse that's knocked out for activin, it does have some alterations. There are more stem cells, for sure. But then as you progress down the lineage, things look pretty normal, right? The subsequent cells in the lineage aren't really elevated very much. And that kind of tells you two things. First of all, in this lineage, the influx from this stage to that stage can't be very high. It seems like this stage is maintaining itself mostly through self-renewal and not through influx from a prior stem cell. And also there must be something else going on here at the level of the stem cell so that it doesn't explode, right? So it doesn't undergo an exponential expansion. Okay, but the problem is both of those situations have big costs associated with them. If you have a very low flux from here to here, then this is exactly the system in which you can no longer have both rapid regeneration and robustness at the same time. So you've just lost that objective by gaining a different objective. And finally, any extra loop we can add onto this renewal of the stem cell that will help bring it back into control should we knock out the activin necessarily eliminates the perfect robustness that we gained by having just a single loop. So then are there ways to work around that? Well, yes, there are. It turns out if you use positive feedback in this system, where something else comes from cells that increases the probability that progenitor cells renew themselves. Originally, I thought this was kind of silly because you have negative feedback, positive feedback, they just cancel each other out, right? But that's not the case because they have different operating regimes. They operate at different levels of cells. And so what you get instead is systems in which you can be both robust and fast at the same time by using positive feedback. And the way that happens is because these systems develop a property that's very useful in engineering called bistability, or switching. That basically, because they're mixing positive and negative feedback, they're stable in two configurations, a high state and a low state. And for the same set of parameters, either state is accessible. Right? So they can either switch into something where they're cranking out a lot of cells or switch into something where they're not cranking out. And either steady state is a stable state. And it turns out that all those cases that are both fast and robust are the bistable cases. And the reason for that is essentially what happens if you knock something out, so that knock out terminal cells so it can regenerate, it switches up into the high state, cranks out a bunch of cells, and then switches back into the low state. Go up to the high speed, go for a while, and then go back down to the low speed. And that happens naturally as a result of mixing the positive and negative feedback. So that's a good thing, right? But there's a price of bistability, which is called hysteresis, right? That hysteresis is if you have a bistable system and you want to switch from high to low, the point at which you switch from low to high is different from the point at which you switch back from high to low. You see that in thermostats, right? Where the thermostat doesn't turn on until it's 72 degrees and doesn't turn off till it's 68 degrees, right? That's a hysteresis loop. And so you might expect, if hysteresis is at the heart of the way growth control systems work in our tissues, that you might actually find even pathological conditions where things get stuck in one state or another. And in fact, if you look, there's all kinds of pathological conditions which have that signature. Things like psoriasis, hypertrophic scarring, cirrhosis, myeloproliferative disorders. Psoriasis is a good example, actually, because there's been some sort of mathematical work on psoriasis. if you're not familiar with this disorder, right, it's essentially an overproliferation of stem cells. It's not cancer, right? These stem cells stop at a different state where there's a lot of stem cells, and because your skin has a way of regulating its overall thickness, when you have so many stem cells, you get a thinner non-stem cell layer, right? And so the thin layer above it, the cornified layer, being so thin is what causes the actual problems in psoriasis, right? You don't have a good barrier. How do we treat psoriasis? In a very medieval way. We just kill cells. We kill the stem cells with things like, coal tar and UV light. And essentially, studies have shown that the effectiveness of treatment is based entirely on how many stem cells you kill. If you kill enough, then the system goes right back down to the original state. Okay, so in psoriasis, there's some kind of immune dysfunction that instigates the switch to the high state, and then you get stuck in the high state and you can't come down. There's a number of other examples of that sort of proliferative behavior. Okay, and then I said there was this other factor that you must have something else that keeps stem cells from exploding when you take away this integral feedback controller. And we spent a long time trying to figure out how you could have control on that without losing the beautiful integral feedback control that comes from this activin loop. And finally we realized that the reason we weren't getting it is because we were looking for a redundant circuit, something that acted in parallel, right, with the control circuit. And what we really need is what's called a failsafe circuit, something that doesn't perturb anything until you lose something, and then it kicks in, right? And it turns out that there is an easy failsafe circuit in this system, which we've known about forever in cell systems in general, but we don't tend to think much about what it's doing, and that's self-feedback of stem cells on their own rate of division. So what I mean by self-feedback on cell cycle speed is what those of us who've done tissue culture call contact inhibition or crowding effects, right? When cells get crowded, they stop dividing, right? We see that in lots of cells, or at least they slow their divisions down. And a lot of recent work tells us that much of that is mediated just by mechanics. If you squish a cell, this transcription factor called YAP rushes out of the nucleus. And if you stretch a cell, it rushes into the nucleus. And that directly controls the speed of the cell cycle. So cells know how compressed they are, and they use that to control themselves. And it turns out if you put that loop in here, you can get results that look very much like we actually see in the activin knockout, which is that these cell numbers expand, but because they're now going slower, the flux that they're putting into the next stage is hardly changed at all, which is why all the subsequent stages are not affected. That's why it's a good control circuit, right? It can even tolerate that knockout of this feedback loop here. But of course, that begs a very interesting question. How would you ever evolve failsafe control, right? Because you only kick it in when something else is broken, right? And activin knockouts aren't going to happen very often in nature. And when they do, nature will be perfectly happy to just let those genes pass out of the gene pool, right? So there isn't a real advantage, it would seem, to having this failsafe control. But if you think about it in a different way, right? Rather than thinking about as how you deal with knockouts organism-wide, let's just think about cell-to-cell variability, right? Cells may just have fluctuations in their levels of receptivity, numbers of receptors, for example, for factors, or they may pick up mutations, right? Cells are constantly picking up somatic mutations. So what happens in the normal case of this feedback, this integral feedback loop, if a cell in here would pick up a mutation that made it less sensitive to the feedback? So now it will only be brought into control if the number of differentiated cells is a higher number than it was before. And so consequently, it won't be in control and it will expand its numbers up and produce that higher number. But that higher number will now feed back on the normal cells, which are not in control at that number anymore. Now they're driven to extinction. And eventually what will happen is even a slight advantage will cause one cell to take over the whole population and give you a somewhat larger number of terminal cells at the end. So that little thought experiment tells you that one of the risks of integral feedback control is it sets up competitions where anybody who steps out of line wins and takes over the population. And this actually has an analogy in the cancer field to a phenomenon called field cancerization, or the cancer field effect, where early mutations spread throughout a population even before there's cancer. right? And so then cancer arises at a high frequency in this large field. Well, that's exactly what this predicts. Any cell that gets a little ahead of the game is going to beat out all its neighbors and replace them and create a field of cells with a mutation. Okay, so does this phenomenon of cells feeding back on their own rate of cell division help with this problem? Right? Do you get some improvement in this fragility here by having it? So it turns out to address that question, we have to do stochastic simulations, basically simulations where we look at what happens in a big population when one cell steps out of line. Okay, and so these are some just individual results from it where a cell steps out of line and instead of renewing itself at a 50% rate, it's now gone up to 80%. Okay, and this cell, boop, fixes in the population. It basically replaces everybody else. Okay, but here's one that renews itself 95% of the time. Basically, almost all it's doing is renewing itself. It goes up to a very high level and then it goes away. So what's happening there? If you look closely, the mutant cell rises, but then the wild-type cells rise. The wild-type stem cells rise even higher than the mutant cells, and they crowd the mutant cell out. Right? So what's going on there is that the mutant cell, by switching to mostly renewing itself, is making a lot less differentiated stuff. The differentiated stuff feeds back on the wild-type normally, so when you take that away, when the mutant cell is growing faster, it makes the wild type expand as well. And all of that increases this crowding effect. But the mutant cell is right in the middle, the epicenter of the crowding, and so it's much more sensitive, in effect, to be crowded out. And so stochastically, you'd predict that mutations that are effectively cancer mutations that should sweep and take over are going to be very frequently extinguished depending upon the renewal behavior of the cell that arises. And this is a little graph showing you that the probability that a mutant will fix is not that high if the mutant just makes itself all the time. The cleverest thing the mutant can do is to continue to make differentiated cells so that the wild-type neighbors don't realize that there's a mutant around, right? So it's kind of a stealth mode. And of course, this is, in fact, the cancer stem cell observation. The observation that in real tumors, at least when you don't look at them at the very, very latest stages, right? In real tumors, the tumors are always making differentiated tissues, right? They're making differentiated cells. They're not normal, but the differentiated cells are there, right? And this would argue that that's because that's a viable strategy for a tumor. Whereas jumping right away to the highest grade in the first step would never work. The tumor would get extinguished by this natural selected for process. Okay, so just to put in some of the things I've talked about in this one tissue, right? I've talked about negative feedback, positive feedback, self-feedback. It turns out there's even more going on. There's another cell type that the stem cell can make, and the same feedback factors feed back in opposite ways on that cell type. And it turns out even that's predicted from the performance objectives of this system, because the kinds of things that feedback systems have to deal with aren't just, getting the parameters right, but also just kind of disturbances coming in from the outside world. It's what engineers call disturbance rejection. And in a simple system like this, if you have, say, something that's killing stem cells or randomly changing the rate of things die, right, it changes the steady state this thing goes to. And the only way to get really good control is to have really steep feedback functions, almost step-like functions. But then that causes these systems to have very unpredictable oscillating behavior. They kind of go out of control the harder you push them to try to achieve control. And that's a structural feature of the system. There's no way to beat that with parameters. But you can beat it by putting a lineage branch in. And it turns out all you have to do is have kind of like a relief valve that allows cells to be pushed out this way when you have too many of them. And now you can get really great disturbance rejection with really high gains in these feedback loops. And in a sense, that gives you a reason why so many lineages actually do have branches. And sometimes, and this is particularly the case in the immune system and the hemopoietic system, the branch goes to death, right? Huge numbers of cells are just thrown away which we tend to think must be because there's something wrong with those cells. But what this is saying is that's just part of a viable control strategy. If you want to control here, you have to have that escape valve so that you can throw cells away in order to keep your numbers constant over here. Okay, so let me just summarize all that from that part of the talk. What I've tried to argue just through these very simple systems that are a little bit complicated but not yet hairballs, that these trade-offs that come from dealing with control problems are drivers of biological organization, right? What looks like kind of kludgy random stuff is actually very sensible engineering when you're trying to have control over size, fluctuations, regeneration speed, and the accumulation of mutations. All of these phenomena, whether you have stem cells, stochastic divisions, lineage stages, negative and positive feedback, multiple feedbacks, self-inhibition, branches, all of those are what you get when you try to achieve this. And you also get other things like benign proliferative disorders, field cancerization, and tumor stem cells. Right? So the idea is by trying to understand the hairball, right, we can have a context in which to understand so many of these phenomena that we actually see. Okay, now the shorter part of my talk is going to move on to a different, but I hope you'll see related topic, which is complex genetics. So now I'm going to step back a little further, and instead of just talk about a single system in which we can identify the players and factors, I'm going to talk about the larger system of how all your genes interact. And that, of course, is a big problem in medicine. It relates to the problem of personalized medicine, right? Most of the important human diseases we face have a big heritable component, right? They run in families, but very non-Mendelian genetics, which tells us that a lot of genes are involved in most cases. If we're going to use genetic information to predict personal risks, we need to know whether genetic variants act in an individual additive way or whether they tend to act combinatorially, right? Because if they tend to act combinatorially, then you have to know which combinations of mutations a person carries, whereas if they act individually, you can just know each one and add up the risks, right? So how additive should we expect genetic contributions to be in the human population? So there have been many attempts to resolve this empirically, and by and large, I would say none of them succeed, right? So we do genome-wide association studies, but the problem there is ascertainment bias, right? All you can find in genome-wide association studies are the additive effects. And in fact, you don't find a whole lot of them in most genome-wide association studies, but that doesn't mean that the effects are combinatorial. It could also mean you're just not looking at the right variants. And so people argue to this day over how do you interpret the fact that most GWAS studies fail to provide a lot of the heritability that people observe. There have been attempts to estimate empirically by breeding, essentially by looking at the breeder's equation and seeing how things pass in families, how much heritability is additive. But it turns out for mathematical reasons, a lot of that is subject to major artifacts that are not controlled for. So we don't really know what the true additive heritability is for most diseases. There's a lot of work on experimental organisms, right, that suggests there's a lot of non-additivity, or epistasis is what most geneticists refer to this as. But a lot of work on experimental organisms involves large effect alleles like knockouts, which are not going to arise that commonly in populations because they're large effect, right? They'll get selected against. So people argue about whether this has any relevance to the human population. And it's certainly true that systems biologists tell us that most biological systems are nonlinear systems. That is, the terms don't interact additively. Here's an example of a typical system from the BioMedNet database, right? And you can sort of calculate the parameter sensitivity. So greater than 1 means a nonlinear parameter. And sure enough, there are lots of nonlinear parameters in these systems. So that would suggest there should be complex combinatorial interactions. But the reality is, with nonlinear systems, if you perturb them only little bits, the perturbation effects tend to be additive anyway, which is another way of saying that any nonlinear system can be treated as linear over some small domain, which is something mathematicians do all the time. This is just an example from that system I showed you on the previous page, where you actually start perturbing the system by playing around with parameter values, but you only give 20% perturbations, kind of like what we think most SNPs in the genome are, small affect perturbations of gene function. And then you see what's the observed change in the output versus what you expected if it were additive. And if you do pairwise, triple-wise, quadruple-wise perturbations, you see it still tends to behave additively, even though the system is combinatorial, right? So all those things tell us we don't have any kind of a priori reason to know one way or another. Are our genes largely interacting in a highly combinatorial way, or are alleles of the genes we have, our common ones, are they interacting this way, or are they just interacting additively? So the question that I like to pose is, are there constraints of the kind I just talked about, constraints that relate to control and robustness, that actually do give us a priori expectations about how gene variants should interact? And there is an interesting set of observations that speak to this question. It's called the theory of survival of the flattest. Okay, and that term was coined by Wilkie and Adami a number of years ago. They were actually studying evolution in computer programs called digital organisms. But it turns out that there are many other examples of this now. What they pointed out is that if you have very high mutation rates, it's actually beneficial to have a situation in which the effects of mutations are made smaller, so that your progeny will survive to carry your other genes. Right? So there's actually an advantage at high mutation rate. Turns out that's probably a very small effect in diploid populations. What's a much larger effect has to do with environment. Okay? There's no real reason why evolution should care to select for genes where mutations don't have a big effect on phenotype, but evolution certainly cares to select for systems where environmental perturbations don't have a big effect on phenotype. Right? That was all what we were just talking about with tissue growth control, is you want to build systems that are well controlled. But the problem is, once you build systems that are well controlled with respect to environmental effects, they automatically become well controlled with respect to genetic perturbations, right? And that's what's shown here, is that as you make things have less variance with respect to external perturbations, you automatically make them have less variance with respect to genetic perturbations. So in other words, genetic robustness builds up. We tend, just by natural selection, to obtain biological systems that are robust to small effect mutations. And that's referred to as survival of the flattest, because the idea is that you're on a landscape, right, a phenotypic landscape. And if you can find a flat spot on that phenotypic landscape, you're better off, because environmental perturbations won't change your phenotype much. But that also means now genetic perturbations won't change your phenotype that much. Okay, but natural selection is far more effective at producing a response to individual perturbations than combinatorial perturbations. And that's just a fact of mathematics. It's the individual effects on genes that tend to drive natural selection. And so that creates an interesting imbalance, which has been predicted to have important consequences. Essentially, as you're selecting for robustness, you're really mainly selecting for robustness to the additive effects of genes. You have no power to select away, right, against the combinatorial effects of genes, unless they happen to be linked, right? And so the prediction then is, as you make things more robust, you make them more epistatic. That is, you build up combination effects by getting rid of individual effects. Now, most of this work has been done at the level of sort of average mutational effect and average epistasis, and we thought that wasn't what was so relevant to personalized medicine, right? What we really care about in personalized medicine are the big effects and how likely they are to be combinatorial, as opposed to how likely is the average effect to be combinatorial, right? We want to know the big crashes, the things we call disease, right? Are they combinatorial in origin? And so we started taking things like this model and various other models from the systems biology literature that are modestly complex and putting it through the paces of this selection for robustness. Essentially, we would find at random some parameters that let the system perform some tasks that we predefined. We'd measure how robust they were to small changes about the parameters, and then we'd just sort of change parameters to see if we could find places that produce the same output, but we're now more robust to the parameters. Basically, we're moving around on the landscape of phenotype till we find a flatter spot. Okay? So here's an example. This is a circadian oscillator model. It can be described in terms of three very nonlinear differential equations. The task it performs, it will be entrained by a light input that's pulsatile to give you fluctuations. And when you turn off the light, it continues to oscillate with the same period and a reasonably good amplitude. That's what your circadian oscillator actually does. So now we take that with the set of parameters I just showed you. It's a hideously nonlinear system. Upward and downward perturbations of the parameters have huge sensitivity factors, this means that a 1 is linear. This means they're quadratically and cubically related to each other, so very big nonlinearities. We look at that system, we make little parameter perturbations, and we say, do the observed effects match with the predictions, assuming additivity, pairwise, more or less, triple-wise, quadruple-wise. If anything, there's some antagonistic epistasis. That means effects are lower than you would predict from adding them up. But we don't see much of this synergistic or combinatorial fragility coming out here. Now we take the same system and we find a different spot on the landscape that's much more robust just by randomly searching. And here's one that produces the same output, but look at how much more robust it is to all its parameters, right? We do the same experiment. Look what you see. So now, first of all, the red is the new experiment. All the effects are smaller because we selected for robustness, right? The system's more robust. But there's this collection of cases that have whopping effects compared to all the individual effect sizes, and none of that effect is predicted by their individual effects. Triple-wise, quadruple-wise, you see even more. This is what these effects look like for the system, right? Instead of oscillating normally and then continuing, they either fail to continue or they have abnormal oscillations and often fail to continue as well. You can play this game with any model system you like. This one, the MAP kinase model, randomly connected model, you always see this buildup of what we call combinatorial fragility, large synergistic epistatic effects emerging just as a consequence of selecting for good performance, selecting for systems that are robust. So, apropos of this room, this phenomenon is what's called an edge of chaos phenomenon. So the term edge of chaos comes from the complexity theory field, right? And the idea is that complex systems often achieve their highest performance in an operating regime just next to the one where everything falls apart. Okay, so it's not an obvious thing to think of, right? But now, if you think about it for a while, there are many metaphors in real life that are kind of like that. Just at the point before everything decompensates, and actually the clinical term of decompensation is a great example of this sort of phenomenon. So just before decompensation is where you're at your peak performance. Okay, and so the reason this is an edge of chaos phenomenon is because we're selecting for the best performance, the flattest. Right? And as a consequence, we guarantee dramatic failures to certain combinations that wouldn't have been so dramatic if we hadn't selected for such great performance. Now, one of the questions that arises in whether this theory really applies to human biology is what happens to combinatorially fragile alleles in a real population? Would they be weeded out by natural selection? So the interesting thing is if you do these kinds of evolutionary simulations, what you get is, in fact, they're not weeded out. They tend to stabilize at sort of minor allele frequencies of 1 to 5%, depending upon what your selective pressures are. So that's the kind of thing that people actually do look for in GWAS, but they don't have the power to see combinatorial effects. And in fact, if you take these toy systems and then you just do like a fake GWAS, right? You make a population in the computer and then you say, well, what if I sampled it and studied it just like a GWAS study? You get all the things you see in GWAS, which is it looks like there's a high additive heritability, but there isn't. It's called phantom heritability. You see that most of the individuals who are affected are doing so by virtue of a combination of risk alleles, and that the variance that's explained by the additive effects is trivial. Right? So that's actually the signature of this sort of high combinatorial behavior. Now, the nice thing about doing it in the computer is you can look in and say, well, what are the alleles that are actually fragile in these systems? Which are the ones that are creating risk? And you can look at them in terms of their flatness in the landscape over the region of which they were selected to be flat. Right? So within that pink region, they're all pretty flat. But which ones are the ones that are combinatorially fragile when you mix them together? It's these. and they all have a common signature, which is that they're flat over the region you demanded that they be flat, but then they start curving up shortly after that. Again, you're sort of on a phenotypic cliff, right? And when you have that, these are the ones more likely, when you combine them, that are going to give you a synergistic effect. In fact, there's lots of evidence that real systems do live near these phenotypic cliffs. Here's an example from some work in yeast, looking at where the level of expression of a particular gene product is relative to the fitness effect of changing that. And it's found to be close to that cliff here. Here's an interesting example from Suzanne Godet and Peter Sorga's work, looking at the variance in expression of things like BAX and BCL2 from one cell to another, right? Because they're not at the same levels, right? There's a variation in a population. And by modeling, asking what happens to the phenotype as you step outside that region of variation, and in both of these cases, there's a huge fitness cliff. Basically, as soon as you go beyond the normal variation, you suddenly get cells that in this case never die, or in that case die too soon. So a lot of biology seems to get perched on those fitness cliffs. And the idea why that creates this synergistic epistasis can be illustrated again by looking at the sort of landscape here. That if this is flat going in this direction and that's flat going in that direction, but if it's non-flat going in this direction, then it's very likely it's also non-flat when you go a little bit farther in this direction. Okay, and so that tells us that one of the ways we can try to make better use of data such as GWAS data is not just to think of every allele as an independent, every allele as sort of its own actor, but think about multiple alleles in the same gene, or about heterozygous versus homozygous effects at a gene as a way of sort of mapping out landscapes along the axes of individual genes and using that to draw inferences about what happens in the landscape in between, essentially in the combinatorial region. Okay, so then wrapping up, in this part of the talk, I tried to argue that a very important control objective, just generally controlling for the impact of environmental variation, buys you the following three features, right? That you build up robustness to individual genetic variation. And in fact, we know this is the case, right? Those of us who've done knockout mice know how often knockout mice look just fine, right? And most genes in heterozygous form give almost no phenotype, right? In heterozygous null form, give very little phenotype. This is not something you see in random engineered systems, right? This can only be there because it's been selected for. Combinatorial fragility occurs for subsets of alleles and is the most deleterious alleles, right? The things that are really going to have selection against them. In the case of human biology, maybe things like neuropsychiatric illness, birth defects, and so on. Those are the ones where you should be especially likely to have a combinatorial as opposed to an additive origin of phenotype. Okay, so just to finish up, I opened up my talk with the question about getting to why, right? Can we get to why biology is the particular hairball it is? Is this complexity really there by chance, or is it necessity? And I guess I'll end with the statement that we may not always be able to find out in every case, right? We're looking at arguments based on plausibility. We weren't there during evolution to see what happened. But to the extent that control is such an important part of biological performance, we have every reason to try to explain biological complexity in terms of the high price that must be paid to achieve good control. And another fact is that because control is so important, right, control makes it very hard for us if we don't have hypotheses about the complexity itself. It actually makes it harder for us to learn things from the data, right? Because control is there to remove influences in the data. And only by knowing that the control is there can we actually go look and see what are often some of the most important players in a system. So I'll just end up by thanking some of the people involved in the work. My center is very interdisciplinary, and we have lots of collaborations with people in different fields. In my lab, the work has been largely done by a student in developmental and cell biology, biomedical engineering, and a physics postdoc who have worked on different parts of this project. And a lot of the work has been a part of long-standing collaborations with Ann Kaloff in the Department of Neurobiology, Anatomy and Neurobiology, Ching Ni in the Department of Mathematics, John Lowengrub in mathematics, Fred Wan in mathematics, and collaborations with John Wilkins on evolutionary simulation and Mustafa Kamash on engineering theory.